Yeah, whenever you're good.
Good afternoon, everyone. Thanks for coming out, and also a big thank you to Dr. Ronholm for making the drive all the way out from Montreal. So just to give you guys a bit of a background, Dr. Ronholm did her PhD here at the University of Ottawa. She also completed a postdoc with the Canadian Astrobiology Training Program at McGill University. Then she went on to do a fellowship at Health Canada, where she used genomics to evaluate the safety of food products. And now she's an assistant professor at McGill University. She works in the Department of Food Science and Agricultural Chemistry. So her talk today will be the agricultural microbiome, manipulating the microbiome of food processing animals to increase food quality. So,
Thanks, Alex. Like I've been telling a lot of people today, you're not going to see a lot of new data today. I started my job at McGill in January of 2017, so it takes some time to start up a new lab. So I'm mostly going to be talking about ideas today, and hopefully some of them are good ideas, but we'll let you judge that. And since it's a student presentation, okay. I'm also going to give a couple lessons that I've learned over a couple generations of doing these microbiome studies, and tell you about some of the things I would have wished I knew about microbiome studies when I did my first one, because I've made a lot of mistakes over the years, but I've learned things from them. And just getting into the generation things, I've been working on microbiomes kind of since like 2012, and we've covered a lot of generations since then. So just out of curiosity, has anyone ever done a microbiome study using VGGE? In here? Couple people. That was just ending when I started. And then we were on to 454. Has anyone used 454 for microbiome work? Do you miss it as much as I do? And then we moved to Illumina. So has anyone done it using Illumina? Okay, a couple people. And I think eventually it's going to be going to long read. Has anyone actually done a microbiome study using like a long read sequencer like the Minion or PacBio in here yet? Okay, interesting. Yeah, someday maybe. Anyways, so I was at a seminar last week, and someone came up to me and asked afterwards, what do you actually work on? What do you do microbiome studies on? And I said, everything. Which is obviously, exaggerating a little bit. But in the last 5 years, I've done the asbestos microbiome, which was really interesting. So this was as part of the Canadian Astrobiology program, where on Mars, there are really big asbestos deposits. And asbestos is a great substrate for microbial life, particularly chemolithoautotrophs, because it's just rich in the electrons they like. So we went to the asbestos mine in Quebec, which is now closed because we don't mine asbestos anymore. But we started looking at everything that lived there, and there's these incredibly rich microbial communities living on this asbestos. And we did temperature studies on them, and what's really interesting is these guys are metabolically active down to at least -5. We never pushed it more than -5, but I think they go even lower. So these asbestos deposits are really interesting in terms of Mars work, and maybe something we should keep working on. I've also worked on endolithic microbes. So these are microbes that actually live in rocks. So these are quartzal feldspathic rocks from the Jeffrey mine, and you see these beautiful layers of color. That's not rocks, that's photosynthesizing bacteria, the different pigments in there. And again, they're living in a rock, but there's these really interesting, rich communities. So my point there is these microbes are living almost everywhere, and they're doing really interesting things. I've also worked on soil. I started moving into clam, oyster, mussel, and other food microbiomes. There's interesting things living there as well, possibly contributing to whether or not these get infected with foodborne pathogens, which kind of started me on the direction for my own research is, can we use the microbiome to get rid of foodborne infection rather than using antibiotics? And that's mostly what I'll be talking about today. So in the last 2 years, I've moved more into looking at the actual mouse microbiota in response to infection, as well as looking at fecal of a lot of food-producing animals in terms of what's there when they're sick and what's there when they have pathogens that could be spread to humans, and what's there when they're totally healthy and don't have pathogens. Oh, and there's a nice little picture from the Arctic, so it's cold up there, I think was my point there. So every time I say agricultural microbiome, everyone thinks I mean something different. So what I mean by agricultural microbiome today is the microbiome of our food production. So what lives in the plants that we're growing to eat, what lives in the animals that we're growing to eat, and what lives in the animals that are producing things that we eat, like milk or eggs. And what association do they have to both disease and food quality? I'm also sometimes talking about their feces, and there's reasons for that. So things that my research is covering in the recent, like now, is we're looking at pathogenic bacteria in the environment, and not necessarily, how they're getting to humans, but how they're spreading AMR genes. Who's out in the environment with AMR genes and who's moving them around? And that's a really interesting question. We're starting to get into microbiomes and greenhouse gas emissions. So there's a lot of interest in, can we actually reduce the greenhouse gas emissions from agricultural operations just by shifting the microbiome? And there's some evidence that might actually work. In addition, we have projects both on meat and on milk. And milk is interesting because there's a lot there in terms of illness. So Staph aureus causes a really serious infection in cows called mastitis. And mastitis affects your cow longevity, cow health, milk quality, and overall production. So it's really important to our dairy industry. And milk also, we're going to get into the vitamin B12 story, and I'll talk a lot about that. But in this case, the microbiome has a really, really clear link to food quality and human health, which is interesting. In terms of meat, there's also a vitamin B12 story to be told. But in this case, other pathogens like Salmonella and Campylobacter could be an issue. And if we could get the Salmonella and Campylobacter out of the cows without using antibiotics, there could be a real benefit to the food quality of what we eat. So when I started this, I didn't know much or anything about cows, but I did get funding to do a mastitis project. So I piggybacked this with a lot of other researchers who are collecting milk from lots and lots of cows for other reasons, and they had a lot of questions about how to sample for the microbiome. And their first question was, how often do we have to sample to actually see a change? I said, I don't know. And I went to the literature, and the literature didn't seem to know either. And then another question was, do you have to sample each quarter individually? So since you guys probably aren't cow people, cows have 4 quarters, and this is an automated milker, which is how we milk cows right now. And you could sample each quarter individually to get a microbiome sample, or you could take it from this little collector here, which is how they sample for things like mastitis. So is there a difference between those 4 quarters? Do you have to sample for them in microbiome studies? And I didn't know that either, and the literature didn't seem to know it either. And then the third question was, what's the effect size? How many cows do you reasonably need to sample to see big differences? And there wasn't, this was in 2016. There's been one paper published since that gets into this pretty well, but there wasn't one when I started. So that brings me to my first lesson for grad students that I really wish I would have known for my first microbiome study, is that have a sampling strategy that makes sense. Sometimes this means doing the pain work that you might not get a publication of, but actually doing a pilot study to just figure out how many samples you need. What's the best way to sample? How do you process your sample afterwards? You're going to freeze it? You're going to freeze dry it? You're going to put it at -20? You're going to put it at -80? And all these questions, unless there's a really systematic study out there already for whatever you're working on, it's not a bad idea to just do that pilot study. So we actually spent some time, and we did it. And it was a really simple study design. So we had 3 cows, and once a week we went in and we took a sample from each quarter, and we did this every Monday morning for a total of 8 weeks. Now, the cows weren't perfect replicates of each other. One was an old girl and one was a young girl, but they were all spread out in the same barn. My barn has 80 cows in it, So you can spread 3 cows out among that barn pretty well and get a good distribution. And when we sampled all 4 quarters, we did all the sterile stuff, like sterilize the teat and strip it first, so you are actually getting raw milk microbiome as opposed to something icky on its quarters. And this produced a lot of really interesting data. So first off, we didn't see that big of a diversity between the 3 cows. Like, this is fairly consistent microbiome data. When we actually analyzed it statistically, there was no difference between the cows for each individual week in terms of AMOVA values. So I've shown you class and order here, and even at the order, you can see it's very consistent. Between quarters was a bit of a different study, bit of a different story. So this is all the cows, all 4 quarters, all 8 weeks, with each of the weeks labeled as different colors. And you can see that the clouds around the 8-week samples are really big here, and the 8-week samples are really big here. Other than that, there weren't statistical differences. And when we got into why this was, this cow and this cow each had a somatic cell count in one of the 4 quarters that was just on the verge of mastitis. So if your cow is healthy, there's not a big difference or a statistically significant difference between the udders. But if one of those 4 quarters is infected or starting to get infected, you start to see that change and you start to see a significant difference. And this was the most interesting finding of that little pilot study. And that's that this is all 4 cows, all the quarter data combined. So you have, it's like you sampled the bulk tanks, but you did it bioinformatically. And you can see that between week 3 and week 4, all 4 had a dramatic shift in their population, which was really interesting. These are spread out completely at different sides of the barn. And I wish I had daily data to see if they all changed on the same day. But within 7 days, they all made the same shift in population. And I can't explain this yet. I overlaid temperature data at one point. It's not like we had a heat spike or anything like that. I tried overlaying precipitation data. It wasn't because of rain. It wasn't because of humidity. My best guess now is that they changed the feed, but I can't prove that yet. We're still trying to dig through farm records and see if we bought different feed that day or that week, which is my best guess. But it's very interesting that all 3 cows had that very different shift on the same week. So my brief little pilot study, basically we don't have to sample from the quarters. The automated milker is fine because there's just not a big difference there, provided the cow is healthy. If any of the quarters have mastitis, you do actually have to go down to the quarter level, or if you suspect one is getting it. Variable between samples or variable between animals in the same barn on the same diet, there's not a lot. They're very consistent. And we really do have to do at least weekly samples. I would love to have daily samples to see if all those cows changed on the same day. So for our mastitis study, we'll be sampling that way. Which brings me to our actual mastitis study. So since you guys probably are all medical people and know what mastitis is in women, it's a little bit different in cows. It sucks in women too, but in cows it costs a lot of money. So from a welfare perspective, in Canada, the most common reason that you'll have to get rid of a dairy cow kind of before its time is because she has an infection that you can't clear up, and this is usually a mastitis infection. And it's estimated that this costs between $5 and $8 million to the Canadian dairy, $500 to $800 million to the dairy industry, which is really huge. So in Canada, dairy farmers don't make money off their cows till the 3rd lactation. The first 2 lactations pay for the cow. So if you have to cull your cow due to an infection before she hits her 3rd lactation, you didn't make any money off of that cow. And in Canada, dairy farmers are running on razor-thin margins, so this is really important to our industry. And it could get worse. We'll see how NAFTA goes. But it's a big issue even if NAFTA goes well. So the threat of mastitis is so great to the farmer's bottom line that prophylactic antibiotics are almost always used when you're drying your cow off. So that means she's done lactating, she's hit her peak, she's going to have a little rest period before she gets pregnant again, and they just give her antibiotics just to keep things cleared up. And there's some debate as to whether this is good or not, like maybe this is making the problem worse, but at the very least, we got to start considering this because antibiotic resistance is an issue. The other thing about mastitis is that once a cow has mastitis in one quarter, the microbiome of that quarter doesn't seem to ever return to normal levels. There's limited data on this. You can see that, it's one study with, I think, 10 in each group. But the mastitic group, which are represented by the red dots, and the healthy quarters, which are represented by the black dots, have very different populations. And you'll see that it actually has to do with diversity. So if a quarter has had mastitis, that quarter doesn't return to a microbiome diversity that you see in a healthy quarter. And if you've ever had mastitis in a quarter, there's a really good chance you're going to get mastitis in that quarter again. So is that because there's still infection in there and it's just latent and it flares up again? There's very little evidence to suggest that. And in fact, when they do whole genome sequencing, it does seem like it's different organisms coming in and causing the subsequent infection. But is it because of that low diversity microbiome that the animal is susceptible to infection again? And is there anything we can do about that? So my second lesson, which I didn't do for my first microbiome study, is basic science. Have a question. And I was telling people today, I review a lot of microbiome studies, and it actually is starting to make me mad that people just go and sequence things for seemingly no reason, and then they think they can publish things. Like, you have to have a question, you have to have a reason for testing things, and you have to have a testable hypothesis. So my first study didn't, your first study should. We're past the point where you can just sequence things. So for this study, we've actually kind of gone in a very obscure and kind of weird sampling design. And the reason is because I don't want to infect cows, and I don't think it tells me anything about natural infection if I just infect cows. I'm going to wait till they become sick on their own, and I'm going to start looking at the microbiome pre-first infection. So was there actually a microbiome problem prior to the very first infection instead of just every infection downstream? Which is, I think, is an interesting question. If we do see microbiome crashes, if there's a reason for that in the barn, or if there's a reason for that in the cow health, this could explain a lot about mastitis. And the reason I think that works is because you very rarely see a cow with 2 udders infected. So is there just the minute little difference between udders that become infected and others that don't become infected? And is that minute little difference exploitable in terms of preventing mastitis? So in this study, we're basically going to collect milk samples from a bunch of quarters, and we're going to archive them. Collect them, stick them in the fridge. And freezer. When they're in the freezer, we're going to analyze the somatic cell count. So somatic cell count is basically how many immune molecules are in the udders, which is an indication of infection. If there's higher somatic cells, there's likely infection. So if the cow has mastitis, we can go on and targeted amplicon sequence all of the weeks prior to her getting mastitis to actually look at what was happening in the microbiome prior to that initial infection. If she's not positive, we're just going to keep archiving the sample till eventually she gets it. So this is going to end up with a lot of freezer space being taken up by milk samples that are just sitting there, because we have 700 animals enrolled at this point in this study. So if you figure 700 times 4 times every week, we're going to have to figure something out for this, but lyophilization is a possibility. The other thing there is that we do actually have 2 really important questions. First is, after we analyze this data, do we get some insight into who is going to become infected down the road? So can we use the microbiome as an early detection system for infection prior to actually getting that somatic cell count? Because once you've got a high somatic cell count, you're not milking that animal for a while. She has to go on antibiotics, and in Canada you can't milk an animal that's been on antibiotics. So having that early detection system would be really interesting for farmers. And then another question is, does the microbiome ever recover post-infection? So the studies that have looked at this have only gone a couple weeks out, whereas the animal might live for another 6 years. Does that udder actually recover in the 6 years downstream? So this study is actually funded for 5 years. So after 5 years, I think we're actually going to get a really good look at, does the microbiome ever recover to pre-infection populations? And then at the end of this, when we kind of think about these two questions together, does low diversity microbiome predispose an animal to mastitis? Is there a specific component to that community that predisposes an animal to mastitis? And can we exploit it? But I think those questions are a couple years off. So this is lesson 3. It took a long time to recruit those 700 animals. So remember, when you're going into a lab and you start a project, your supervisor has done a ton of work on that project before you start it. And I don't think I ever appreciated that as a grad student. So my next story that we are kind of working on right now is vitamin B12. So out of curiosity, is anyone here a vegan and they're willing to share that information? Wow, that's crazy. At McGill, it's like 50%. Okay, no vegans here. Does anyone know what vitamin B12 is really intimately and worry about it at night? Okay, vegans worry about B12 at night. They're really well versed in what this is because it's a problem for them. So vitamin B12, well, vitamins in general, are something that you need to survive that you don't make. So you have to get it somewhere else. And we kind of realized this was important when we started making germ-free animals. None of these animals survived. Because there was no microbiome to produce a lot of the vitamins, and sometimes microbiomes help you in producing vitamins. Vitamin B12 is a really specific vitamin that has a really specific structure that I'm not going to get into, but in the human gut, it's made, but you don't absorb the stuff you make. You only get your vitamin B12 from your diet. If you drink milk, that's mostly where you're getting your vitamin B12 from, but you do get a little bit from meat. So if you're a vegan and you don't drink milk and you don't eat meat, you really have to think about this and think about supplementing or where you're going to get your vitamin B12 from. As of 2011, 4.6% of Canadians were deficient, and this might seem like a lot, or it might not seem like that many, but it's actually, it's a big issue because you suffer from anemia and you can also suffer from cognitive impairment, which is probably really significant to all of us. But the problem with this is if you look on milk, if the vitamin B12 is listed in the milk, it's listed as, missing some on my slide there, it's listed that a glass, it should be right here, but 46% you get from a 250 milliliter glass of milk. That's not true. That's an average. That glass of milk can realistically have anywhere from 16 to 57% of your daily intake. So if you're only getting 16 from that glass of milk, you're not getting enough vitamin B12, and it would be really beneficial to have dairy advertised as a good source of this and a good consistent source of this, but it's just not true right now. So we could fortify it, but that adds cost to the dairy industry, which is already on razor-thin margins, so we're probably not going to do that. And the synthesized version isn't as bioavailable as the kind you actually get from bacteria and archaea. So what do we know about vitamin B12, and why is it so variable in milk? First off, we have to give the cow adequate cobalt or it doesn't synthesize vitamin B12. Cobalt's needed for the actual structure of the vitamin. If we make smaller silage, which is what we feed the cow, there's no effect on the vitamin B12 production. So this doesn't really do anything. We can supplement it with probiotics, but there's not really a big apparent difference in the concentration of vitamin B12, even when you stick Saccharomyces in there. If we add detergent fiber, we can get a modest increase in vitamin B12. And this is interesting to people because at least we have some way of doing it. And it's the first evidence that what the cow is actually eating in terms of silage could actually be affecting the vitamin B12 concentration, but we're still missing the mechanism. How does what we feed it actually convert into the production? And I think the answer here is probably the microbiome. What you eat affects your microbiome, which is in turn going to affect your milk output. But we don't actually know who's participating in this process at the moment. So I was telling some students today that when I was sitting here falling asleep in the lecture room, once Kathy Wright, who just retired, said that we really have to remember that not all people are inbred mice, which is a really good thing to remember, but I think it's something we sometimes forget. And I think another thing to remember, especially for my research, where people's like, oh, that'd be so useful in humans. Not all humans are cows. So in terms of their biology. And it's really true with vitamin B12. So in vitamin B12, we actually do make it. But we make it in our large intestine and absorb it in our small intestine. And if you're familiar with people, the large intestine comes second. So you're not actually absorbing what you're making. In cows, they do it the opposite way. They're synthesizing in their rumen, and then they're absorbing it in their small intestine. So they actually do absorb all of the vitamin B12 they're making. They take their own for whatever they need for their own nutrition, and then they secrete the rest in their milk. So it makes a lot of sense that synthesis of vitamin B12 by a certain microbiome, then chewing it up by other parts of your microbiome is what's actually affecting the vitamin B12. And we know a lot about this. We've just never put it all together. So we already have a list of all the bacteria that are really good at making vitamin B12. It's probably hard to read, but there's a list. It's very complete. So our question here is, those guys that are making vitamin B12, are they more common? Are they at a higher population in the rumens of animals that have a higher vitamin B12 synthesis output? And that's one thing we're going to be looking at. We also know the pathways to make vitamin B12. You can synthesize it kind of de novo, or you can grab other structures that look similar to vitamin B12 and kind of salvage it and take it through and put it into a vitamin B12 structure. So are these synthesis genes, in terms of the metagenome, not just 16S anymore, are they more common in animals that are actually putting out a lot of vitamin B12? Now, if that was the only story, it would kind of be really convenient for this, but it's not. Bacteria also really like to eat vitamin B12. Some make it, some eat it, some do both. So there was a really interesting study in humans where they gave people tons of vitamin B12, 1,000 times the recommended daily dose, and no one was secreting any more in their feces than they would have on a normal dose. So the idea is that the bacteria were all eating it in their guts. So this paper raised a question that maybe vitamin B12 deficiency isn't so much a dietary problem, but it's a microbiome problem. If you have a microbiome that's consuming a lot, maybe you're going to be deficient. So that's a question that still has to be answered, but I'm not touching that, so that's a good question for somebody. So we do know that bacteria eat it. So are the bacteria eating it faster than they're making it in the rumens of cows that aren't producing a lot of vitamin B12, or are they eating it faster in the feces? And all good questions. We also know the routes that it's used for uptake, so it's really convenient that we already know a lot about how bacteria deal with this vitamin, because it'll be really easy to analyze metagenomics data. And we know the genes that consume it as well. So we can do metagenomics to look at the actual gene content in terms of, are we eating it in the intestine? So our study design by this time is probably really obvious, but we have 70 cows. We're going to sample, well, we already did. We sampled them all twice. And we're going to look at vitamin B12 concentration in their milk, in their blood, in their rumen, and in their small intestine. And we're also going to look at the microbiome composition of each of those. And we're going to look at some metagenomics as well. And I was telling some students today, they think their work stinks, or they think their E. coli stinks, or their lab stinks because of their E. coli. Wait till you smell a rumen. These things are disgusting. It's the worst thing I've ever smelled in my life. So you actually have a fistulated cow. You guys may have seen that on TV or on a thing, where you actually like pull a rubber plug out of the side of the cow, and you can stick your hand into the rumen and get a sample. Worst smell of your life. But someone had to do that 140 times for this study, so I hired an undergrad. And he worked for his money, but he did a really good job. He was a good sport about it. So this is where we are now. We have a nice freezer full of rumen and feces and milk samples and blood samples, and we have a couple undergrads who have to, these are all freeze-dried rumen samples. They're in a freezer because I'm scared of things. They don't probably have to be. But when you rehydrate these, they smell like rumen again. So we've got 2 more undergrads. Oh, and I lost a picture there. This picture was just showing that the vitamin B12 in the milk we collected from those 140 samples and in the blood we collected from those the variability was amazing. So we do have really, really low vitamin B12 producing animals. We do have really, really high vitamin B12 producing animals. So I think it's actually going to turn out to be a nice study at the end. Which brings me to my fourth lesson. Be afraid of the kitome. And I was going to say be very, very afraid, but some nights I don't sleep because of the kitome, and I want you all to sleep at night. So be the right level of afraid of the kitome. Control for it, and always run these generous donor samples, but sleep at night. So that's what we've started doing, and this is one generous donor over 2 different kits, and this is the other generous donor over 2 different kits. So if you do run these generous donor samples, I mean, as long as your kit's okay, it's not scary to run them. I've talked to people who are too scared to run them. Don't be too scared to run them. It'll tell you if your kit's contaminated. So the other thing that we're just starting up now, which I hope is going to be kind of the trademark of my lab, is antagonistic interbacterial interactions. So bacteria like to fight a lot. So in my head, kind of in the early stages of infection, you have your enteric pathogen coming into your small intestine, and before that pathogen can really set up shop, they have to make room for themselves. Like the intestinal microbiome, there's a lot living there. There's competition for nutrients, there's competition for space, and if you got a lot of virulence genes in there, maybe you're not the best competitor in the world. So we're finding more and more, and there's more studies building on this now, that yes, colonization resistance plays a role. So like C. difficile, it's just the space issue. It's not an actual competition issue. But now we're seeing that some pathogens can actually kill off, not just for space, but the actual competitors that are going to take them out specifically. So there's a couple studies on this now, but I'm a Listeria person at heart, so I like this study the best. So Listeria lysine S is this thing we didn't really know what it did. But we found that when Listeria was expressing it, and not all Listeria express it, just the particularly virulent 4B strains express it, it alters the gut microbiota. And it was kind of unclear about why it did this. So this study came out in 2016, and it's really nice. And it found that it was specifically targeting Alloprovitella and Allobaculum. And it was cutting these levels really, really low when this gene was overexpressed. And it was cutting them, pretty good when it wasn't being overexpressed, but being expressed. Whereas if you put a Listeria in that didn't have a Listeria lysine S, there was no effect on Alloprovitella and Allobaculum. They expanded this study and actually showed that Alloprovitella is something that's really good at producing acetic acid in the gut, which inhibits the growth of Listeria. And Allobaculum produces butyrate, which doesn't prevent its growth, but prevents the expression of virulence factors. So it's specifically taking out the bugs that are producing things that could make it less of a pathogen. And if it's overexpressed, you do get a deeper infection. It goes further into your actual organs. And you read this paper and you're like, seriously? Is biology actually this nice that it's actually that targeted and crazy? But the paper, they've done follow-up studies, and it all appears to be true, which is kind of amazing. So that covers, the pathogen being hostile to the commensals, but there's also commensal hostility towards the pathogen that we've now found. So this is in gram-negative, so a little bit of a different story, but type 6 secretion systems were fairly recently shown to be involved in interbacterial competition. So they're things they use to take other bacteria out. And they've actually found this nice little story where Vibrio cholera has this first type 6 secretion system that comes and it's contact dependent. It's actually a little syringe that injects a poison into neighboring bacteria. And he injects E. coli dies. So he's really good at killing E. coli. But he injects P. aeruginosa. P. aeruginosa gets mad, expresses a second type 6 secretion system, not this one, and actually kills the Vibrio cholera. And they have videos of this. I didn't show it today because they never really work in classrooms like this, but Google it sometime. It's amazing. You can see them turn on and fight each other, and then you see the Vibrio cholera just slice. It's amazing. But I think a question they failed to answer in their study, as amazing as this study was if you have this specific strain of P. aeruginosa in your gut, can you get Vibrio cholera? which, I mean, that's a good question. I would love to see that study. But can we find more of these interactions? And can we actually exploit them so that, you know, take this antibiotic, or take this prophylactic prebiotic or probiotic to get rid of your Vibrio cholera infection? Like, is that something we can do to get rid of antibiotics? So we've made basically no progress on this. This is just an idea. But we decided to work at Salmonella enterica as our model system to study type 6 secretion systems in these kind of competitions. And we have a lot of reasons for that. And the first reason is that there's 4 known type 6 secretion systems in the genome of Salmonella enterica. But when they found these 4, and they're not in the same Salmonella strains, they're kind of spread out over the phylogeny of Salmonella. And if you know a lot about Salmonella phylogeny, it's big. So there's a big diversity of type 6 secretion systems in the diversity of Salmonella. And when they did the search where they found the 4, they did that in 2009, and they had a whole 44 whole genomes available to study to find those 4. Today, NCBI has 7,502, and the Cystomics project, also out of McGill, has 4,500 really, really nice genomes. So we're in the process of kind of sorting through these 4,500 genomes from the Cystomics project and looking for new type 6 secretion systems in Salmonella that we might not know what they do yet. But SPI6 is kind of the most well-studied one at this point. It's capable of killing Klebsiella in the gut, but doesn't seem to be capable of killing E. coli in the gut. So we do see that specific interaction, but I don't think we understand the range of this particular system yet. So what else does SPI6 kill when it's in the gut? And does anything fight back are kind of the first questions we're going to attempt to answer. So I will be recruiting a PhD student for this particular project in 2019. At the moment, Salmonella is interesting in mice in that it doesn't always do predictable things with the microbiome. So we're actually going to take a year and kind of pilot our mouse population and really figure out what our mouse population looks like in terms of microbiota before we start our infections. So we have one PhD student working on that right now, but hopefully we'll get a second by the time we start to actually look at what Salmonella does. And if anyone's interested, I'd love to hear from people who know stuff about microbiome already. Okay, and I told people that I was going to take the last couple minutes to kind of rant and rave. And I hope no one minds about that. It's just when I'm sure some people in this room actually are starting to get papers to review, and there's so many bad microbiome studies that find their way into the literature. And this is a quote. Rob Knight is actually the senior author on this paper. Everyone probably knows Rob Knight. But scientific advances based on microbiome studies depend on carefully planned, well-executed, controlled, and reproducible studies. And just going out and sequencing things are not any of these things, which is what so many people are doing right now. So don't be one of those people. I was one of those people. You don't want to be one of those people. So know exactly what you're doing and know exactly why you're doing it when you're doing science, but microbiome studies. And a lot of the times when I review papers and the authors get back to me, they're saying, well, this guy did it. That is not a good answer. That is never a good answer. Just because it's published doesn't make it right. Know what you're doing, know why you're doing it. And one of the issues I think with 16S right now that people are struggling with is that there's a lot of choice. You really just have to pick 2 primers, send it to a sequencing center, and tell the sequencing center which primers you want them to use. And then you're done. You have your sequences, you can analyze them, you pay someone to analyze them. But do you know why you chose your primers? And lots of people have well-justified reasons for using the primers. I'm not saying my primers are the only ones that are right, but I use 548 and 806. And the reason I use these 2 primers is because they produce an amplicon that overlaps completely on a MySeq. So if you understand how the MySeq works, you have your microbiome, you amplify your amplicons, and then you stick them to a glass chip, if anyone's ever seen a MySeq chip. And when they sequence, it's not a black box, it's not magic. It's amplifying the DNA on the chip and producing a light signal, which is then taken a photo of, and the machine does a whole lot of things and spits out your sequence. but it all comes down to these photos. So there's a limited length that these can get, or else they start curling back on themselves, and you're not going to get the resolution in your sequencing. So right now, Illumina has a cycle 600 kit, and they have a cycle 500 kit, and that's mostly what people use for MiSeq. HiSeq is a different story. But people have complained about this. People have complained about this directly to Illumina, and I know Illumina's response is, what's your point? But the cycle 600 kits start failing at a certain point long before the cycle 500 kits do. So there's a lot more error in this region. Error is bad for 16S. 16S relies on having really accurate sequences and not having error recorded as a novel OTU. So really, you're limited to 250 base pairs if you want a low error study. And sometimes you don't want a low error study. I've read studies that are awesome that had a lot of error, but they just wanted a long amplicon, and some of them have justified reasons for doing that. But most of the time, you're going to be limited to 250 base pairs. So these two allow a 239 amplicon, which fully overlaps. When you get your data back and you do that whole assemble contigs thing, the full overlap is going to use the p-value to correct any error. Whichever read is the best is the one you're going to get, which really reduces the error in your sequencing. This gives you a lot of high-resolution, high-quality data. If you don't have that high-resolution, high-quality data, you're going to have more error. And if error matters to you, this should matter. So I have read studies where they've actually said, like, this produces more diversity than this. And people like submitted this for publishing, but if you calculated the error based on the Illumina having a 1% error read, all they were seeing was error. The numbers worked out exactly. So the studies that compare different sized amplicons and claim that one is better than the other because they provide better diversity, it's just error. And those studies are published. So I think another thing to think about is metagenomic follow-up. So when I kind of designed each of these studies, I had everything done in 16S to get a look at who's there and see correlations. But if you want more detail beyond the correlations and a little tiny peek at the mechanisms, you really have to do the metagenomic follow-up. So I've kind of structured all my studies so that I am going to do metagenomics on 5 to 10% of my samples to really get an idea of what are happening in the unique ones. Metagenomics is interesting in that it's really hard to bin sequences and get whole genomes out of metagenomes. But this paper is coming out in a couple days. Watch for it, Nature Biotech. It's not mine. I'm just a fan. But what they've done is they've used the 10x chromium, and this is normally used in cancer studies. Maybe some of you guys have heard of it, and they're using it now in vertebrate whole genome projects. But this paper has actually used it to look at metagenomes. And what they're doing is they're taking the DNA out of these mixed microbial communities, and they're size selecting it, so you're getting big chunks, maybe not whole chromosomes, but big chunks. And then you partition it in these oil droplets and perform an isothermal amplification so that each of your big chunks of DNA has a unique barcode. Then you use this unique barcode to actually put whole genomes together from your metagenomic sample. And it's turning out to be better than, we had binning techniques, we had the synthetic long read sequences. This is turning out to be better, and they've actually showed that really nicely. So I think this is going to change things in terms of non-culturable bacteria and their contributions to both biology, cycling, everything really for the non-culturables. And I think it's going to be really interesting for the movement of antimicrobial resistant genes. I don't think we have any idea about how non-culturable microorganisms are moving AMR genes through our environment. So I think this is going to be good for that. So that's my talk for today. Most of my team just started, but Julian, he's working on the vitamin B12 project. Soyun and Adam are both part of the NSER Create in milk quality, and they're both working on the mastitis projects. And Ziwi and Maria just started working on the interbacterial interactions. So they've done all the work the little bit of work I've showed you today. They did it. And I think that's it. If anyone has questions, I'm more than happy to take them.